Maybe should we introduce each other or ourselves briefly, or for the moderators? So my name is Nils Ghelenborg, and I'm an assistant professor at Harvard Medical School in the Department of Biomedical Informatics, where I lead a research group that is focused on visualization for all sorts of biomedical data, ranging from molecular data all the way to clinical patient level data. That's where I'll leave it for now.
I'm Peter Sorger. I'm also a faculty member professor at Harvard Medical School in the Department of Systems Biology, and my lab is a member of the HTAN group studying precancer atlas on melanoma, and we also work with an OHSU team on breast cancer.
My name is Jason Swedlow. I'm at the University of Dundee in Scotland. I run the Open Microscopy Environment Project. I serve as an external program consultant for HubMap.
And I think the purpose of the current, so the organization of the session here is going to be a brief plenary session right at the moment, and then we will split into 3 breakout groups and then come back into plenary at the end. And what we thought we would do is, as we go into the breakout groups, the subgroups from this, we will have people individually and briefly introduce themselves. But for the moment, Jason, Nils, and I thought we would just go through and tell you quickly what the 3 sub-breakouts are, which you can you'll see on KI Storm, I think, in a minute. And then you can decide which one to join. And then we're going to come back at the end in a second section of the plenary to come back and actually talk about practical steps towards integration across programs and projects. So we're going to tackle 3 separate topics having to do with data and visualization, and then we'll come back and think about how those can be brought together. So breakout session 1 is going to be in visualization and headed by Nils. Do you want to say a word about that, Nils?
Yeah, sure. Thank you, Peter. So for the first breakout session, what we do want to talk about are sort of overall, like we're thinking about all the many data types that we have to integrate when we're working with Atlas data, and the need to build new visualization tools that are capable of this kind of integration. So this spans imaging, this spans other data types like sequencing-based data types, mass spec data types, etc. And so we want to talk a little about how can we get it started? What should integration look like? There are many different ways how visualization tools can integrate different data types. We want to talk a little bit about sort of really, what do we need to do as a community to get together and develop visualization tools rather than doing this on our own? How can we maybe build components that can be mixed and matched, etc. And finally, we want to think a little about, what are the major use cases for data visualization. There are obviously a lot of different visualizations that are needed for Atlas data, for CellAtlas data. But what are the critical ones? And in particular, we can also think about which are the ones that have already been solved, about 1 million times when you think about scatter plots for all sorts of embeddings versus those problems that are much harder to address. And think a little bit about what could be a roadmap to accomplishing the design and development of these integrated visualization tools.
The second breakout session is the one that I'm going to lead, and that's going to be a search across metadata, data, and coordinate systems. So we were tasked with thinking about how search is going to work, and the most obvious type of search is across metadata elements, tissue, disease types, perhaps measurements, and we'll want to look at that, or clinical features. There is going to be a technical issue with how we do efficient indexing. But then I think there are more interesting types of search we'd like to think about. How would you look at features, for example, if you have image-derived features or specific types of enriched gene sets, how would you actually do that across different types of tissues and different data modalities? So this type of sort of integrated cross-functional search has the kind of semantic element that hasn't really been defined, and we thought we would try to explore that from both a set of requirements and also from a set of technologies.
And I'm leading the third breakout, and this is asking the question of how can we analyze the distributed data sets that are definitely a feature of our current world. So how do we find those data sets, browsing them, accessing them, and start integrating them, what tools exist for that, and how are people actually working in practice? Is that offline, various types of cloud resources, et cetera? So not to take more time, I think one of the things that we wanted to do in this document, if you look under the top green box under the plenary part, we made a start, and I would emphasize this is a quick start of the different types of data that we want to, that we're going to need to think about across these different domains that we're talking about in the session, indeed, across the various projects that we're running in. So Nils and Peter might want to add to this, but I think what we wanted to do here is ask the participants to start adding types of data into that first plenary part green box. So if there's, we had a certain, we started off with a bunch of imaging data types and some omic data, but I'm sure there's many other data types that people here would want to add to that. So in some sense, knock yourself out and fill that box up.
Indeed. That would be
so I think editing's already begun on that. We seem to have lost proteomics, but I imagine whoever is doing that anonymous otter will put it back in a moment. So we're not going to belabor that, but that obviously document remains open despite the discussion, and then we'll come back to that again in the final plenary session. So unless there are any questions about this or other comments from the three discussion leaders, I think we're actually in a position now.
Just remind us when to come back to this room.
Yes, we'll let you know. The idea here is that this next section, we're a little ahead of time here, but we now have 75 minutes in the next session, and we will call a break roughly halfway along.
Okay, so they're working in 3 separate groups straight into the second, straight into part 2?
Yes, so you'll now see those on KA Storm. What is 17A is integrative data visualization with Nils. Search is with me, and analyzing distributed data sets is Jason. And then we come back for a final, I believe it is 30-minute plenary session at the end, which has its own set of tasks, which is to really begin to imagine integrated search and visualization. So to begin to put a conceptual frame on what would we want at the end of the day, and then begin to think about how that could be achieved. Our goal was to go back and actually now try to imagine what a sort of kind of integrated search and visualization engine would look like, and how common that is across the areas. And I was wondering if you thought it was helpful, Nils, just to go quickly through, we'd have to do it relatively briefly, but in 2 or 3 minutes, what we learned from each of the breakout sessions. Those are largely summarized in documents, but we could probably say a word to that.
Right. Yes, I could try to summarize what came out of our conversation. What I wanted to add to your point about what we want to do now, I think, yes, integration of search and visualization is an important aspect of this. But the other thing that we wanted to talk about is, like, assuming we have some sort of a reference or some synthetic images or something that we could use to link together data sets, for example, or to look at, variation between a reference and a sample, how we would accomplish this. Because one of these how might we questions that came up in our conversation was how we created integrated visualization between spatial data and non-spatial data, in particular when they're from different samples. And so for that, we really need to solve this question of reference systems and reference coordinates. So that would be another important point to discuss in this context of this larger breakout, because it also applies to search, etc. Okay, is that second group or that third group back as well? Looks like we have about 40 people in the room, which is
fairly apologies. Yeah, Sorry, Niels, we were running a little bit behind, but yes, we're all coming back now.
Great, no problem. So Jason, I don't know if you heard what Peter and I just discussed here. We thought it would be good to maybe do a very, very brief summary, and it's probably a bias summary with a bias on or towards the things that are relevant to the big group discussion, which would be about integrating search and visualization, and I guess also analysis. We should acknowledge that, and how a reference would be used in the same context. How do we bring references into visualization, search, and analysis? To I guess to some extent, how would we create these references? Did I summarize this correctly? And Mike had another great question for me. I will answer in private. It's
because we have relatively limited time. In order to have some discussion, Nils, we should probably just very, very quickly fly through, the 2 or 3 highlights of the sessions that we each had, and then get on to the sort of question at hand is how we'll pull this together. Did you want to say a word about the joint visualization that you haven't already, or?
I will. So the main point here is for us, we've talked about a lot of different things, but in terms of like the more actionable things, we were wondering, can we, first of all, identify a data set or assemble a data set to use for visualization tool development that really has a lot of different data modalities in it, and that would be coming from different consortia. So that was one question as sort of a foundation for visualization tool development. And then we had a very, very basic, sort of question about, could we build a joint, or collaboratively build a visualization tool that can be used to integrate spatial data, like say imaging data, with some non-spatial data. And ideally for a use case where these 2 data types may be from the same tissue, from the same part of the same tissue, but from different samples. So there's no direct linkage between the 2. How would we accomplish this? So those were some of the big questions. There was another, I think, very nice how might we question that sort of came up early in our conversation. Like, how do we even know what's already out there? There are so many different assays, and they are sort of coming out of different communities, and they might already have tools, but there's no good overview of like Atlas visualization tools. That's my summary.
So from the search discussion and an integration discussion was also quite lively, and we were trying to look at how we would look across metadata types, data types, and coordinates. And I think one of the things we determined was actually this is a fairly hard problem that's been given relatively little study. And we were reminded that even extremely useful data sets like ENCODE don't necessarily as yet have very sophisticated search functions in them. And that search functionality is hard to develop in the absence of defined use cases. And we need to know what it is, what kinds of things people would want to search for. So our primary recommendations, in a sense, for going forward is that there needs to be some level of search, but that could almost be at a kind of Miami level, across some fairly simple metadata fields against controlled ontologies. That's the easiest to imagine. There's a lot of enthusiasm for spatial search, so searching against spatial features, although how that would be accomplished. And that an important thing going forward would be to get the relatively limited number of people in all of these programs who have real expertise in search, indices, ontologies, semantic concepts together as an expertise, and then feed back to the leadership of all of these efforts what's really realistic with respect to an actual integrated search function. So I think there was some concern coming out of our session that this is imagined to be, it's going to be imagined that you could do arbitrary search against all fields and all features of all atlases, and that is going to be exceptionally difficult to achieve.
Thank you, Peter. Jason, do you want to give this a try too for your group?
Sure. So first of all, thanks to a very fun discussion over there in analytics. And we talked about several things, but our discussion was guided by, okay, imagine we wanted to take 2 data sets and do some sort of querying across them. when we did reference some of the really impressive studies that were presented at, in the plenary by the Lung Map Consortium. So, okay, you have data from different parts of some organ. How would we actually even, that are different and distributed, how would we bring them together? And that, as you can probably anticipate, got us quickly into formats, et cetera. And I'll just, full disclaimer, those of you who know me, I didn't do that. So definitely started thinking about concrete ways that we could build capabilities to make those different distributed independent data sets comparable and connectable, especially through a CCF. I have to highlight that it sounds like we had several different similar discussions, I think especially that Nils mentioned, I guess also Peter's group, about, okay, how would we find out what's out there, et cetera. Kati Borner highlighted work, I think, in the CCF breakout in terms of listing data set, at least the data sets and the tissues that are out there. And in my memory, trying to remember the breakout I was in yesterday, definitely came up just a survey of what exists in the different consortia across the different communities is across all of our different tooling, protocols, tissues, et cetera, is probably, it's probably time for that, because it seems like it comes up over and over again. We need to understand what's out there. We concluded with a target call. So if you want to read more about this, and I think Nick Nystrom will bring this up in the plenary, hopefully. We have a target call that we think we could develop a proposal for. That's in the notes, in case anybody's interested.
Great, thank you, Jason. So, did anyone want to add anything to that? Nick, did you want to, that's for the later plenary or for this breakout plenary? I'm not sure.
That is for the soapboxes for later.
Say again?
That is for the soapboxes for later.
Yeah.
Okay. Okay. Great. So I think, well, we have a bunch of things here, but maybe to loop back to this question about the one thing that I think is very tricky to deal with in terms of visualization and mapping things, and I guess also querying things is, I mean, there are technical challenges, but I think in terms of the business logic, like how do we, incorporate reference information into these tools? I think from a visualization point of view, it is, incredibly helpful to have like something to compare your data to. And I'm wondering, because I mean, a lot of visualization tasks are actually to find differences between two things, right? And seeing those differences is often easy once you know what to compare against. And this having this reference to provide context for what you're looking at is really critical. So I'm wondering if people have thoughts on that. Like, are we even close to being able to come up with like agreeing on a reference? I mean, there have been suggestions in our breakout, we can of course try to just use a bunch of samples and agree, well, that's somehow what we're going to use as a reference. But I'm not sure if that's really what everyone would agree on. So I would like to hear some thoughts on that. Like, can we come up with a reference and then we can talk about how we could use that, both for search and visualization. And
Nils, one thing that for us was undefined, and there wasn't, we noticed that yesterday there was a session on common coordinate frameworks or reference frameworks, was what actually the underlying sort of dimensions of such a thing are. In some cases, I think people have interpreted as spatial dimension in the same way, radiology is fitted into pictures of the human chest or something like that. Yeah. In other cases, it's a cell type census. So it's an enumeration of cell types, and the cell types of the reference frame. So I think even in some very fundamental way, the dimensions of these common reference frames were unclear to us, but they will define data integration and search.
Absolutely agree. There are a couple, at least I just saw some people who are involved with common coordinate frameworks on the call. Is anyone still here who is working on common coordinate frameworks?
So I can say a few words. So
Thank
in
you.
some instances, you might have a person with a healthy lung, and that person volunteered to do a lung biopsy. And we might also have a CT scan of that lung. So in this case, you register the biopsy sample inside of the person's lung from which those samples were taken. This is a very unusual and typically a very nice case to have, but this is not normal for HubMap, definitely not. And so then if you don't have the same organ from one person from where also the tissue samples were taken, then you need to have a consensus organ or some other reference organ or some other approach, for instance, the vasculature one, which we are exploring, to actually refer back to. Now, let's assume we have a consensus organ, just like what the Allen Institute is doing for the brain, for the human brain. Then you can, of course, register back your biopsy or other data back into that reference organ, and you can do this for 100 samples, and then you can spatially search for those tissue cuboids. Initially, these cuboids are registered on the gross anatomical level, so this is very rough. As soon as you have the biomolecular data coming back, you can do a biomolecular registration, and there are wonderful machine learning algorithms which actually help you to warp as needed and put one dot pattern back into the reference dot pattern. And this way you can register it at a much finer level. Now you have the registration, and you could do that across consortia, and then you could do this in anatomical space, and we are working on browsers there. And then also with Nils' team, we are working on a tissue viewer, which you can then also use to overlay these data sets over tissue samples. But you could, of course, also create a semantic space, a TSNA or UMAP or other space, and you can couple these two spaces. And then you could select a cell in one space, and you would see where it is in the other space, or you select a cell in UMAP or in an ontology, and you see where it is in the anatomical space. In order to make this all work, you need to agree, as you already pointed out, Peter, on the ontologies you're using, on the terms you're using. You need taxonomies. And you need that not only for cell types, but you also need it for micro and macro anatomical terms. And that's where these anatomical structures and cell types tables come in, which you might have seen yesterday in the CCF session. And so we have tried to see if we could get these ASTC tables, anatomical structure cell types tables, for 5 to 10 organs, and see if we all can use them. And I think that would be a huge step forward for all of us. Ultimately, you also want these 3D objects. You want these 3D objects annotated with those terms so that you can use collision detection to automatically annotate tissue pieces.
In that circumstance, how does one deal with even in normal tissue, one thing we were wondering, how does one deal with fairly strong inter-individual variation? Or, once you get particularly down to cellular resolution, the gut was the example we were discussing, every crypt is sort of a little different from every other crypt. So, we either have, what we have now is hand-drawn pictures, like the anatomist did in 1930, which was actually generally pretty spectacular, or we have things in UMAP, but how we would associate the two was sort of still fairly unclear to us.
So there are different approaches right now. One approach we are exploring is to use the vasculature. So again, if you go to the meeting notes for the CCF from yesterday, you will have papers there and also more figures on how that might work. It's a very abstract space again, but you can fold it back into the human body ultimately, and it will fold back differently if it is your body or my body, and some parts will be missing because you're male, I'm female. I think this can be done, but it will take data, which we currently don't have within HubMap in sufficient amount. We will need to understand much more about human diversity, and we also need data at the single cell level.
Peter, you're muted.
So, but I'm going to
interesting to think how that actually would work in the case of a disease as well, right? I mean, so I think those of us working on tumors are particularly struck that it'll be very interesting to see it, but I think it even comes in the normal at what spatial scales you'll have conservation. One's recollection of anatomy is that you have conservation at gross tissue scale, and then you have it at local scale where you have repeated structures, and in between things get a little more amorphous. You'll have more or fewer of any particular structure, more or fewer alveoli or something in the lung. Sorry.
And I think it makes a lot of sense to focus on what is clinically and health-wise relevant. And some things might not matter, but other things matter increasingly. So at the functional tissue unit level, which we are proposing as a way to focus on initially, I think we all might have very similar glomeruli. We might all have very similar alveoli, not if you smoke, but so I think it will be very interesting to identify what's similar and what's different. But again, we need data across many, many individuals to learn more about that.
You know, one thing I wonder if this came up elsewhere, and I should let Jason and Nils or anyone else pitch in, is I think in all of the discussions I've been in is because people who were in our discussion will have heard this before, because we're very vertically organized in these sessions, we have to go from collecting data to getting it out the door. These very critical key technologies, such as building these reference frames and having common ontologies or controlled vocabularies around them, is actually relatively weak. So we have relatively weak integration across the different programs. There's some, I think this Human Cell Atlas has more simply because it started first, but a lot of the others, I get the sense we're all in parallel trying to tackle the same problems. And there may be, for something like search and visualization, it may be very important to actually now work with our programs to build strong links between them. And that practically means resourcing them in some way, because these are all computational challenges, and that is already pretty under-resourced in most consortia.
Just pick up on a couple very quick things. A theme that we talked about a few different ways, and I'm repeating myself, but is to look beyond our specific breakout into what was done in the COVID studies that we heard about from LungMap, into the various reports of cross-consortium capabilities. That seems to be very important coming up over and over again. So I'm just restating what you said, Peter. At least to identify what's there, and then to source that. And then the other aspect, this goes back, I think, a little bit to what Nils was saying, but also to the question of variation. We all know those things exist, and there's going to be no substitute in sourcing that data and beginning to measure that variation and working with it and get those examples in our hands.
Just jumping on here to remind everyone that in about 5 minutes, they're going to move us over to the main plenary.
Okay, so we have 5 minutes left. Do you think we can come up with something that we could propose as a collaborative effort? And I think Peter articulated very well, what some of the challenges are, and it's really, I mean, if you want to collaborate on like tools like search or visualization, there is actually some other work that needs to happen first. And that work is very hard to pull off because of how these consortia are set up. And although, and I should point this out, there are some efforts across consortia that already are in place, such as a cross consortium curation working group or Cell Atlas curation working group that has been meeting for almost a year at this point, at least to exchange information about how people are tackling the various problems they're facing in the context of metadata collection, curation, et cetera. And I could imagine that similar working groups, and I hate proposing another working group or another meeting, could be put in place to actually, but to tackle very specific problems. And I would like to see that to happen, at least in the context of visualization. I think the problem that we talked about in our breakout, how can we get together and build something together with a very simple deliverable If we can make that happen, at least we have a foundation for future work. I think we shouldn't try to solve the hard problem first. I think we should really do something very basic and maybe bring people together who can deploy a visualization tool in the context of at least 2 or 3 of these data repositories that we're building right now. So I think that is a really important outcome, for me at least, for this meeting, is that we can get together and do that. And I think there is a very clear overlap between what HCAN is trying to do and what HubMap is trying to do, because both of these are very image heavy. I think with HCA, there are also imaging assays, as I understand, but can we identify the right people and get that going? And in that context, we will have to address these issues regarding what is the reference system, what are the APIs, and I hope that sort of has a bigger impact. That's something I would like to see happen somehow, and I'm happy to put some of my time into that. I think if that's a kind of a soapbox that we want to have later, I think Peter and I communicated a little bit about that earlier. I think that would be really good as a result of this. But Peter,
Nils, one thing that came up in the previous session that I was in, Atlas Integration had the same thing, is that we're going to suggest to to the supporting agencies and to platforms, we're going to need a resource, an effort. It's both, I think, technical and curatorial to actually find out what is in these various programs and follow up on areas where they're very key technologies that are actually quite tricky to deploy and could be at least inspired by each other. There was some skepticism about direct code release in our session. But in addition, just to find out what people are doing in such a fast moving field is just not going to happen by waiting for papers to be published. And I think that was one of the key things, is we're going to have to actually resource it. I don't think it's a huge resource commitment, but I think it's one that spans all the various projects.
Yeah.
So I just put us in for a soapbox on that, and we'll see if we're chosen.
Cool.